What is the difference between reducing sugar and non-reducing sugar? So the reducing sugar can reduce the other and the oxidize by itself, mean reducing sugar that has capability to release the electron and reduce other. So that's why reducing sugar can give the test with Benedict solutions. For example, we have glucose and fructose and galactose. So these all are monosaccharide, and these are reducing sugar. While in the disaccharide and all polysaccharide are reducing sugar instead of maltose. So the maltose is a disaccharide, but it is a reducing sugar. So we're going to check that what is the difference between reducing sugar and non-reducing sugar. So the contrary of this, non-reducing sugar cannot reduce the Benedict's reagent. So it is converted into the monosaccharide, and then it will give the reducing sugar test. So first, we are going to apply the Benedict solution test on reducing sugar. Now, if you're going to check that the Benedict solution, so we have reducing sugar plus the Benedict solution, which will release the copper sulfate, that will release the copper plus 2 oxidation state, and this copper. So after 90 degrees Celsius, this reducing sugar will release electron, and that will reduce the copper. And this will change from +2 oxidation state to +1 oxidation state. And the sugar, when it will release the electron, it will get oxidized by itself. And in this way, this color will change first from green to yellow, and then orange, and then red. So it means that that is the reducing sugar. But if this sugar is not giving results, there are two possibilities for that. Number one, it is not the sugar. And number two, it is non-reducing sugar. Now, how we can test that? So if there's a non-reducing sugar, so we can use hydrochloric acid to break the bond between disaccharide and polysaccharide by the process, the hydrolysis. So we add disaccharide sugar, which is non-reducing sugar, plus hydrochloric acid, and after hydrolysis, this will break the bond and convert into monosaccharide sugar, and now it will be a reducing sugar. Now we will add that the sodium hydroxide. Why? Because in the presence of acid, Benedict solution cannot work. To neutralize this acid, hydrochloric acid, we are using sodium hydroxide base to neutralize this acid for the working of Benedict solutions. So then we will add the Benedict solution and increase the temperature to 90 degrees Celsius, and this will give us the result brick red precipitate. So if it's a giving of result in a sequence like green, yellow, orange, and lead to the brick red, it means it was non-reducing sugar. We converted into the reducing sugar by breaking glycosidic bond with the help of hydrochloric acid. But in some tests, it's not giving the same results. What does it mean? It means it was not the sugar. So that's why after this procedure, this will remain blue solution. That means that's the negative results. Now, on the basis of this concept, we are going to solve some question. So we got a concept of the Benedict solution result, like if we have a copper sulfate. So now this copper sulfate with blue color, if we dissolve it in the solution, that will produce the two type of ion. First, it is going to produce the copper plus ion with the +2 oxidation state, while the sulfate ion with a -2 oxidation state. Now this copper ion, so after the heating with reducing sugar, that will convert into +1 oxidation state, and it will reduce. Now, on the basis of the Benedict solution test, we can get two informations. Number one, so the sugar is reducing or the non-reducing. And number two, the concentration of the sugar in the solution. For example, if the result is coming blue, it means that there's no sugar present in the solution. So, but if the result is coming green, so it means that low concentration of sugar is present. And if the result is heading toward the medium, the concentration like that is yellow, it means medium concentration of sugar is present. But if after heating the 90 to 100 degrees Celsius, and this result is going toward from orange to red spectrum, so it means that's a high concentration of sugar is present. So on the basis of Benedict solution, we can get the idea, the presence of the reducing and non-reducing sugar, and the concentration of the sugar in the solutions. So on the basis of this concept, we can solve the AS biology, the MCQs questions. So this type of questions are possible in our exams. So in this question, the examiner has given us the 4 different solutions and these different results. For example, the 4 solutions were tested with the Benedict solutions. The table shows the color of the solution after testing. Now, the solution number 1, the result is green. Solution number 2, the result is blue. The solution number 3 is the brick red, and the 4 is the yellow. Now, the examiner asking which row show the solution that could have given these results. So there are the 4 rows are given. So these are the 4 options, and we have to select the correct option according to our concept. Now, what the concept we got? So the concept is that the lowest concentration of the sugar solution will be green, and it will give a green color after reducing, and the highest concentration will be brick red, and the yellow solution will always be in between of the green and the red. Why? Because the concentration of yellow is a greater than green, and this concentration is lower than brick red. So in this way, this concentration will be in between of that. And we will check that in which option, if this concept is being followed, that option will be correct. And second idea we got, if there's a color is coming, the green, there will be some concentration of sugar. But if there is a color is blue, it means it is non-reducing sugar. So we're going to check the first idea. We are going to start from the bottom. So here we have the first is the D. Now in this D, the solution number 1, it should be the green. And he's saying like 0.05% non-reducing sugar. So if it is the non-reducing sugar and this concentration is 0.05%. So, but if it's the non-reducing sugar, this color should be blue. But in solution, he is giving green color. So that's why the solution number 1 option is wrong. We're going to move to the C. And in the C, so here's saying like solution number 1 is a 1% reducing sugar. That's fine. It's giving green. Now the solution number, the 3 is 1.5% reducing sugar. So what is our idea? So the solution number 4, which is yellow, it should be in between the green and the red. But when we see that this concentration is not coming in between green and red, so that's why that is wrong answer. We're going to move to the next option B. And in this option B, we shall check a same concept. Now, the solution number 2 is correct. Why? Because 0% is giving blue color, but solution number 1 is 0.5%. Solution number 3 is 1%. So the solution number 4 should come in between the solution number 1 and 3. but it's not coming in between. It's showing less than solution number 1. So that's why these solution row are correct. Sorry, that is the B is wrong way. Why? Because 0.1% in a solution number 4, it is less than solution number 1. It means that the examiner is saying like the yellow color concentration is less than green. So that's why the B is a wrong answer. We're going to check that the A answer. So obviously it's remaining is A, and if you're solving in the examination and you have a less time, so then if you find your answer first, that fine, well and good. If there's a one option is left, so no need to go for that further and apply the correct and go to the next question. So we're going to check that how it's going to be the correct. So now in this option, if you see that the solution number 1 is 0.05%, and solution number 3 is the 1% reducing sugar, and the solution number 4 is coming in between solution number 1 and the solution number 3 concentration. So that's why this answer is correct according to our concept. We're going to move to the similar type of another general question. So which carbohydrate give a brick red color when boiled with the Benedict solution? Now, here, examiner has given us a different type of the carbohydrate. For example, cellulose, fructose, glycogen, sucrose. So there are some polysaccharides are there, some monosaccharides are there. Now, what the concept we have? We have this concept, all monosaccharide are reducing sugar. So if they are the reducing sugar, so they will give the result brick red with Benedict solutions. So what is the monosaccharide here in this question? So in this option A, cellulose is a polysaccharide that's non-reducing. Glycogen is a polysaccharide, is a non-reducing. Sucrose is a disaccharide, is non-reducing. While the fructose is a part of a monosaccharide, it is our reducing sugar. Now, on the basis of this, we got an idea, all monosaccharide and in a disaccharide maltose are reducing sugar, while the instead of maltose, all disaccharide and all polysaccharide are non-reducing sugar. So we can get the idea. So the reducing sugar will give positive result with the Benedict solution with the sequence of the green, yellow, orange, and red. Hope you get an idea. Thank you. Thank you very much.